Hi, my name is Cameron Williford, and I will be walking you through my research on surveying COI primers to estimate biodiversity in environmental DNA. Biodiversity refers to the variety of life that is present on Earth and can specifically be applied to an ecosystem. Within these specific ecosystems, all species leave traces of their DNA within the environment, and that DNA can then be used to identify what species are present. That process is called metabarcoding. Metabarcoding uses DNA to identify taxonomic groups, thus allowing us to target freshwater aquatic insects. The identification of species is reliant on the matching of the sequences with reference to their barcode, which are in open source databases. Specific primers are used to target the standard barcode region of the genome for identification, which is the mitochondrial cytochrome oxidase gene, or shortly known as the COI, in animals. However, quantifying the biodiversity using environmental DNA is often hindered by the specificity of the primer that is used because different primers differentially target different groups of organisms. Therefore, we tested several sets of published primers to determine which would help us best target aquatic insects within Southwest Virginia. The two pictures are taken from the cited literature which the tested primers are based on. Primers are short DNA sequences that match the genome and flank that section of the genome that is being targeted. We chose several sets that were determined to be best for targeting insects. Environmental stream samples were taken from Wildwood Park. We vacuum filtered the samples through cellulose nitrate filters with pore sizes of zero point 0.2 and 0.45 microns. The zero point 0.2 filter size was used in hopes of capturing more DNA within the sample, while the larger pore size allows us to collect more water because it does not clog as easily. The goal is to capture as much DNA as possible. A polymerase chain reaction, or better known as a PCR, was ran in order to amplify the targeted region of the genome so that we can sequence it. This was done using a thermal cycler, which is seen on the right-hand side. Primers in the PCR determine which region of the genome is amplified, and in this case, it is the COI. Here is a table that summarizes the combination of primers we use to accomplish this. Note that the primers DG and JG are less specific. For example, they have a Y in their sequence, which allows it to pick up either a C or a G base within DNA. A PCR gel was ran in order to show if the PCR was successful. The image shows that our PCR was successful as clear bands are shown. However, completing the PCR gel, we noticed that the combination of BR and BF primer showed no bands, therefore we did not sequence them. A PCR cleanup was done in order to purify the DNA and remove any excess primers, enzymes, or any unwanted reaction components that were left behind. The first set of primers, named MiCOI plus the HCO, had the highest percentage of overall data that were insects as seen in this chart. These efforts were used to develop proper methods that will be used to develop baseline data that will be used to compare the biodiversity before and after the execution of the Mountain Valley Pipeline, which is a natural gas pipeline more than 300 miles, which is planned to go across thousands of bodies of water across West Virginia to Virginia. This combination of primers will be used on all the stream samples from Southwest Virginia. Additionally, we have mock DNA sequencing data to analyze the use of these primers to see whether we can infer species relative abundance using sequence data in relation to the biodiversity before and after the pipeline installation.